Good afternoon, everyone, and welcome to our third and final Science for Success webinar this spring. Our last topic will focus on fixation and sulfur fertility in soybeans. But before we move into that content, we'll just give a brief introduction to Science for Success for those of you who are just joining this webinar for the first time in the series. Science for Success is a United Soybean Board supported project that brings together soybean extension specialists from across the United States to collaboratively deliver best management practices to U.S. soybean farmers and other soybean stakeholders. And we do this through conducting collaborative research trials, some of which you'll hear about today. And we also do this by leveraging our historical knowledge from previously conducted soybean research and being out in the field for many years to sort of amplify our local extension impact up to the national level. So we are thrilled today that one of our Science for Success team members from Kansas State University, Dr. Ignacio Campiotti, will be facilitating this webinar. But before we move into the content, we'd love to know what states the attendees on this webinar are from. So we're going to put up a poll and ask you, what state are you from? Fill this out, and we use that data in a variety of ways. We'll give you a little bit of time to fill this out. And with that, I will turn it over to Dr. Campiotti to go ahead and kick off the content portion of this webinar.
Thanks, Rachel. Thanks for the introduction. Let me see here. Want to make sure, are you seeing the first slide? Good, excellent. So as Rachel was saying, let me go back to the first slide. This is an effort that has been implied in multiple states. So we have here all the states available. So, I mean, and then the presentation in this case on my side is mainly focusing on nitrogen fixation and sulfur fertility. So at the end of the presentation, we will have also Eros Francisco from Auburn University and Sean Castile from Purdue University that they will be joining us to discuss and provide kind of a questions answer session on this topic. So going through, I mean, the initial phases on this project, I mean, I need to acknowledge, I mean, all the work being done by our graduate research assistant, Luis Felipe Antunes Almeida. He's our master's student at this point, working on the project. And he was the one behind coordinating, collecting data. So we'll be presenting some of the initial results on this nitrogen fixation map that we are building for the US, and also the implication of sulfur fertility. So, and also want to acknowledge a postdoc from our team, Adrian Correndo, that is also, I mean, another one behind the scenes working on mainly basically trying to put some sense on all the information and providing some kind of a new insights on this data. So, but I will not focus only on that project. I will try to provide a brief overview of mainly of a few different topics that we have been publishing, just to give an overview of what is a nitrogen fixation and what is the overall nitrogen demand for soybeans. We'll discuss a little bit about effects of genotype, environmental factors on nitrogen fixation, even some questions that we have in the past connected to the effect of fertilization on soybean crop. And then we will stop on what are the implications on management practices. Can we do something from management to help or boost that fixation? The last part of the webinar will be focusing again on mainly just sharing all the results of our first publication on the yield response to both nitrogen and sulfur. So let's go ahead and start with the nitrogen demand. So we will go ahead and then main point, and I would like to start from the basics. So I mean, I would like to start showing this figure. This just relates the amount of nitrogen that the plant needs and different crops they need in terms of, I mean, grams of carbon that is fixed via photosynthesis, and then overall the grams of energy, you know, that we have on the seeds. And you will see that in terms of amount of demand of nitrogen, soybeans is by far one of the largest, I mean, nitrogen demanding crop connected to the other crops that we always, I mean, usually grow in the US. I mean, corn, sorghum, Soybeans is by far with high protein. That is basically one of the main aspects that is connected to the high nitrogen demand. And then that is one of the things why, I mean, nitrogen fixation is becoming a big actor role for soybeans as a main crop. It's one of the crops that requires amount of nitrogen. And economically, if we need to apply fertilizer to soybeans, it's completely impossible. So fixation plays a big role in terms of satisfying the nitrogen demand. And nitrogen per se plays a major role in the physiology of the crop, mainly on the seed production and on the protein content. And we have different sources. I mean, you already heard this term of fertilization, but when you're looking at plant demand, if you think about basically compare with other crops, you mainly have what is coming from the soil and what is coming from the fertilizer. For soybeans, in this case, is basically what is coming from the soil. The other big component that in many situations is a little bit unknown, how much nitrogen is coming from fixation. So we talk about this biological nitrogen fixation. And then we know that the plant tried to modulate and basically tried in many situations when the demand is not met, is really trying to remobilize, moving that nitrogen inside of the plant. And this is a process that we call remobilization that usually happens towards the end of the season. And the plant usually do this process in terms of satisfying the amount of nitrogen that the seeds need for reproductive purposes. So, well, if you look at fixation, and usually we focus on this, I mean, I'm guessing that if you look at this picture, many times you will not be able to see a picture like this, where you see a plant that is on the left under fixation, I mean, without fixation, so you see that yellow pale nitrogen color, and the other situation when it's with fixation. We usually don't tend to see so much this situation, but nitrogen deficiency could happen. And in situations that when nitrogen deficiency happens, it's mainly connected to a problem with the nodulation process. If the nodulation is not well established, nitrogen fixation is not functional, and then the plant usually will start showing these deficiency levels not early in the process, because the amount of nitrogen needed for soybean early in the season is small. But as we move into mid to late summer, we will start seeing that these plants will start showing up. And this will be an indication that fixation, nodulation was not well established early in the season, and fixation is not functional. And as I'm showing on this kind of a small schematics on the side, you will see that the nodulation is a process that is established on the roots, established early season. So we will show some studies that we also conducted to see if we can recover. But in many situations, it's important to understand that this is a process that established early. And then what happens inside of the nodule is just a transformation from atmospheric nitrogen all the way to a reduced form that we call here the ureates. The most important aspect is to understand that there is a highly reductive nitrogen that is atmospheric nitrogen into the nodule, and then there is an exchange. So this process is not free. So there is an exchange that the plant basically releases carbon to the nodules, and the nodules will release nitrogen to the plant. And that's what we call that there is a symbiosis. We'll discuss a little bit about application of fertilizer and nitrogen specifically. In many situations, based on our research, we see that we don't see a big effect. And then, as mentioned before, it's uneconomically, but we will be getting into this point in the next slides. So we did many studies, I mean, in looking at fixation. But like it or not, it looks like one of the topics less investigated, more if you are thinking about trying to understand how much soybeans are fixing. And as I mentioned before, here are some pictures, I mean, that we conducted many studies. This was one in the greenhouse. So you get to see in a kind of a zoom view soybean roots, and then within the soybean roots, you get to see the nodules. And then in a sectional cut, you will get to see these pink colors, and usually are basically showing that the nodules are basically more functional. So one of the first studies that we conducted was trying to understand that fixation and how much is coming from fixation. So here I'm showing just a simple relationship between soybean yields and the total amount of nitrogen that is within the plant at the end of the season. So imagine that you are going to the field before harvest and you look at the entire plant. So just to give you a simple number, so if you look at soybean yields, that could be around 60 bushels. On average, you might have inside of that plant 300 pounds of nitrogen. And then the question is, where is the nitrogen coming from? And when you start thinking about, I mean, from 300 pounds, how much is that coming from the soil? How much is that coming from fixation? There is a portion, very relevant portion in many situations, in many soils, that this nitrogen is coming from soil. The other aspect that we always tend to emphasize all the time, and we show these envelopes here in red, is that the demand of nitrogen is never constant. Usually we say that soybeans need around roughly 5 pounds of nitrogen per bushel. But if you look at these two scenarios on the envelope, there are situations that we have less inside of the plant, and there is a situation that we have more inside of the plant. And in many of these scenarios, it's because the plant tend to dilute nitrogen, and sometimes they tend to accumulate more nitrogen. So requirements in terms of many nutrients for crop yield and demand is never constant. And we need to try to understand exactly how these requirements are changing in different regions and different soils. And then there was a question, I mean, when we discussed about this study, that was connected to the partial balance. And partial balance, I mean, the way that we are seeing here is very simple. If I'm fixing 50, 60% of my total demand, and if I'm removing 70%, my balance in many situations might be negative, which means that I'm fixing less, which I'm removing. So we start kind of investigating all the research being conducted on this aspect, mainly focus on without considering root contribution. And then what we start finding is a situation like this one. So I will try to keep it simple. You will see here different lines. The lines are showing all the observation from many different studies. And there are 3 different groups. The blue group, the blue line, are situations where we are fixing a lot of nitrogen. And in many situations, those are soils that they are sandy soils, low organic matter. And you will see that when we're in that group that we are fixing a lot of nitrogen, our balance in many situations, I would say more than closer to 40% tends to be positive, which means that soybean is helping to leave nitrogen in the system. Let's go to the red group. Is what happens usually in many cases. We are around 58% of fixation, the rest of the nitrogen coming from the soil. You will tend to see that in many situations, the amount of positive nutrient or nitrogen in the soil leaving after soybeans is small, 15%. And in situations where fixation is poor, it doesn't work really well, this green line shows less than 30% total fixation from the demand. And you will see that we have almost no positive balance, which means that in those scenarios, soybeans are removing more nitrogen that basically are contributing by fixation. Someone might say, why is this relevant? If you start thinking about sustainability, and if you start thinking about what happens for my following crop, this could be one of the things that we need to start thinking about. If I'm mining, if I'm removing more, that the crop can fix nitrogen, we will start seeing impacts on the following crops in the rotation. And then we try to do some analysis very briefly here, is just to say, is there any relationship with the budget? I mean, with this balance and the yields? And we see, yes, there is a relationship only in situations where we have poor fixation. So the green line, if you remember, are situations where we are less than 30% fixation. As we are increasing yields, our balance is becoming more negative. In many other places, there is no really clear relationship between yield and fixation, which means that we can have very large yields, I mean, closer to even 100 bushel plus. And in many cases, we can have a lot of variability. So we can have situations that we have some positive value, positive here, and then some negative total budgets. So that's why it's important to try to understand how much is coming from fixation. And that was one of the main ideas I'm trying to think about why we want to look at this aspect, because there doesn't seem to be a clear relationship between fixation and yields. And we cannot assume that in many situations, if I'm yielding more, the amount of nitrogen that I'm leaving behind is very low. So we start looking and studying the fixation zone. And then the main thing that we wanted to understand from the beginning is what is the variability of fixation and how does it change during the season, the growing season. So here is a theoretical representation on the fixation, the proportion of nitrogen fix. And you will see we start from planting time. Usually fixations start a little bit late. I mean, we start usually around B4, B5 with 4 or 5 leaves. And then you start seeing basically how it starts growing and growing the fixation over time. We are reaching a maximum peak that usually is when we are in full seed production. And then after that moment, the seed becomes a very strong source for all the carbon in the plant, and then that fixation starts to go down. Okay, so that seasonal fixation is increasing over time until a peak, and then it starts declining over the season. And we have a study that we start looking at, okay, let's quantify how much nitrogen is coming from fixation. So this was one study that we published a couple of years ago, and we were looking at in many situations, I mean, we are reaching 50% of that total fixation around the moment that we start forming pods. So, and then what is interesting is that we have a lot of variability in the seed filling. So at the end of the season, we have situations where if the health of the canopy is bad, and if there is too much pressure, we start seeing that that fixation starts declining. If we have a very good conditions end of the season, good precipitation, solar radiation, and temperature, so we see that the plant can continue that to satisfy that demand. And that's why we have so much variability on the contribution of fixation. And if you want to see it in a different way now, looking at more the proportion in percentages, you will see that on average, in many of these scenarios, we are closer to 50% of total fixation. And again, the time for maximum contribution of NDFA in this case was around R4. So very similar to what we mentioned last time in terms of, I mean, basically leave it closer to the end of pod formation. And if you think about the time for 50% of the maximum was really getting into the late vegetative or early reproductive. So it's important to understand this because we are seeing that in many situations, once we are reaching the maximum, and depending what is that maximum, that doesn't change. So we need to find ways of boosting that fixation and increasing basically the contribution of nitrogen coming from this process. Here is another study that we did last year, but in this case, we wanted to identify, as you mentioned before, if you remember one of our first slides, sorry, the relationship between uptake, and in this case, is the relationship uptake and biomass. And why it's important to understand the relationship between uptake and biomass is because we wanted to understand if we can use, in many situations, the amount of the plant growing and growing vegetative stages, I mean, as proxy of the fixation. So, and we are seeing that as the plants are growing and they are basically accumulating more vegetative tissue, more biomass, I mean, we are seeing that the plant needs more nitrogen, and this black line is very linearly. The questions are, where is that coming from? Is it coming from the soil, and is it coming from fixation? In many situations, we tend to see that early season, there is a high contribution of nitrogen of most of that demand that is coming from the soil in combination with the process of mineralization. And then later in the season, we really start depending much more on the fixation. And that's why getting into the end of the season into a good canopy health condition, it will be very relevant to maintain the demand. And if you look at the demand for nitrogen does not stop. And this is the only way that we can basically continue to increase yields. So the nitrogen fixation, that contribution increases with the biomass too. And there are many factors. So if you wanted to understand what are the potential factors connected to help boost that fixation, we look at a study, we look at the amount of the duration of fixation, how much the duration, the fixation continues during the season. And we were seeing that we can increase the duration of fixation as we increase the season length. So in many situations, as we plant early in the season and we have more days, those plants can grow more, and then we see some correlation with the length of the season. So in many situations, when we are thinking about double cropping or late planted situation for soybeans, we might see those late planted or double crop soybeans that they have lower fixation. Stress conditions, any stress condition, we talk about heat and drought, any stress will be impacting first, in many situations, the plant. And once the plant is sensing, then it's impacting basically the nodule and the fixation. So any stress condition that is impacting the plant and impacting the ability to basically continue growing for that plant, it will impact the fixation. And lastly, if you want to connect with soils, the high organic matter situations, when we have basically even high clay, because it's nitrogen that is being protected, sometimes, I mean, having a high organic matter and high amount of nitrogen coming from soil also makes a negative impact to that fixation too. We did a study, we look at the genotype, we tested different genotypes. I will not get into this, but I mean, our main idea was to see if there was any change on fixation across the different decades. So we went to really all genotypes, and then we test like different technologies, and we didn't see much of an effect on fixation, which means that we haven't seen that we improve fixation or we improve the contribution of fixation over time, but we haven't seen also the opposite, that we were basically have a negative impact on that. What we are seeing all the time is that most of the fixation changes with the soil type and with environments. And here is an example, right? It's an example showing what happens to fixation in different years, and you will see that it's never the same. In many situations, could be a linear phase, increasing during the season. In some situations, it's more like a plateau. But in many cases, it really depends on the amount of nitrogen coming from the soil. So depending when we have that basically peak of nitrate mineralization, and if it's too early in the season, we start seeing that basically that delays the contribution of fixation. If it's in the middle of the season, we will see that basically still affects the fixation because that level of fixation is not achieving the maximum. So understanding and trying to put some numbers in terms of the impact of soil is one of the big factors that is still quite unknown for the fixation. Just to finish some of the things I wanted to mention from this introduction quickly was, we also start thinking about what happens if we look at fertilization from corn and what is the impact on soybean yields. So if we tend to over-fertilize corn, do we see an impact, a negative impact on soybean yields? And this is a study that has many, many years. I mean, has been done in Kansas, and then what we are presenting here is just soybean yields. The treatments, I mean, the different colors are different fertilization rates for corn, and you will get to see that even in high fertilization rates from corn in the previous year, when we are looking at the soybean in the following year here, we are not seeing effects. So no effect on soybean yields, I mean, from even situations that we tend to over-fertilize corn or even under-fertilize that corn. Similar study we did in our locations, and we tend to report the same situation. So it doesn't seem that we see a negative effect on the previous crop that is moving to soybeans. And same on the budget. So we calculate basically in different ways. If we have a positive, more nitrogen leaving behind, do we see any effect? And then again, in many situations here, we're presenting like the historical data on the case one and the study that we did in that location. And in many situations, even in situations that we are leaving more nitrogen in the soil, we're not seeing a negative effect on soybeans. So now let's talk about a little bit about what are the things that we can do to help fixation. So we have a few different projects that we conducted to look at late inoculation was the first I'm presenting here. We also will discuss very quickly about the co-inoculation. And we try to think about the concept, can we inoculate soybeans? Can we get into soybeans late in the season? So we did a study looking at late inoculation. We look at basically inoculations in soil at different stages after planting time, before and R1, apply liquid close to the plant, and to see if we were able to change the yields. I mean, the summary of this slide is basically that in many situations, we are not changing any yields. So we are not helping to improve in many situations. And the early season inoculation was the best. And we haven't seen also any potential impact, only in a few sites on the quality side, on protein or oil concentrations. We haven't seen much of effect also on compared to the traditional inoculation. So basically having an inoculation at planting and making sure that in locations where you don't see any effect, or you don't have any soybean growing previous in those regions, I mean, it's important to have that inoculation being done early as possible at planting. We did a similar study looking at co-inoculation. There were many questions about connecting broadleaf rhizodium with Azospirillum in this case. We did many studies in many locations on this co-inoculation to see if we can have a growth effect. And what we are seeing very little effect in many situations. If you compare all the treatments with only Azospirillum or broadleaf rhizodium or no inoculation, we see that in many situations, in situations where we have soybean growing before, even without inoculation, we have the same yields that with the co-inoculation. So now let's go to the study that we have conducted the last 2 years. We basically put together data from year 1, and then we are basically now conducting all the studies for year 2. Many partners across the state looking at this contribution of nitrogen and sulfur. And the main context, we have 26 different locations across the US for year 1, where our main idea was to make sure that we look at yield and trying to start quantifying what we call the uncertainty. What is the error around the yield estimation? We look at a protocol that is the same across all the locations, which I think that is one of the big benefits on trying to look at these multi-state regional studies. And we took a look at very different factors, yield, fixation, and our main goal was, again, make sure that we quantify what is the overall uncertainty around this yield response. So these are all the locations, geographical distribution. You will see that we have studies across the entire US, unique protocol, 5 fertilization treatments. We will get to those fertilization treatments. We have a control, no nitrogen and sulfur. We have a nitrogen treatment that is only 30 pounds, 30 kilos added at planting. We have a sulfur only, a second one that is 34 kilos, I mean, planting. And then we have a mix of both. And we have one that we use as a full, which our main goal was to see if we are limited or we are seeing some limitations of nitrogen in those situations. So, and we have half of that in the nitrogen at planting, and then the other half was applied all at R3. So remember these 5 treatments, we were using this and showing the results from the first year. We measured fixation. That was one of our main measurements in terms of quantifying the contribution of nitrogen coming from nitrogen fixation. If you look at yields, and remember the treatments, no nitrogen, no sulfur applies. Our gray situation here, the check, nitrogen only, then the yellow is sulfur only, green is nitrogen, sulfur, and then the purple is the full. And you will see, almost regardless of the locations, you will see that in many situations we see very little response, except for many of the studies conducted in Indiana, and Sean can comment more on this. I mean, that they have more responses to sulfur. We have seen some responses in other places. So we try to classify more the yield response in this idea of what we call the non-responsive. We have no response to any of the treatments, and some of the responsive, where most of the time we see the response coming from sulfur and also from the combination of nitrogen and sulfur. And you will see that we have the non-responsive, kind of a light red, and then the responsive in kind of a light blue. We also try to quantify the nitrogen status of the plant. So we start using this concept that is the nitrogen nutrition index. As the plants are growing, the plants are diluting nitrogen. Early season, those plants, they have high concentration of nitrogen. Late season, as the plants are growing, they have lower concentration. This shows the changes in concentration as the plant is growing. And we try to use this slope to quantify scenarios where plants are under deficiency below this line, and their sufficiency or in luxury all the time that they are above the line, okay? And this was the main idea. The main goal was to see if we can correlate, or at least present this scenario of when you look at the responsive versus not responsive, and here we're showing all the different treatments. We tend to see that in the responsive situations, we have more observations, I mean, higher frequency of the observations under deficiency. So they are showing a little more nitrogen deficiency. And this is important because if we are trying to start developing our recommendation, we need to develop first a diagnostic tool. And we are moving on the direction of knowing when we need to recommend an application. And this is the goal of recommending a diagnostic tool for. So in those scenarios where we have lower NNI, which is means that we have the efficiency, most of those scenarios were connected to yield response. And when you are asking yield response to what? Well, mainly in many situations, if you look at non-responsive, there was nothing. There was no response in terms of yield. Here, the nutritional gap shows a difference on what it will be the yield response. And then the main point to emphasize here is for the responsive sites, their yield response in many situations, in more, this nutritional gap is connected to sulfur. Okay, if you look at the relationship or the observations on nitrogen, we don't see really a clear response. But if you look at the sulfur application, the sulfur application shows that as in many situations, in those scenarios where we're applying more sulfur, and we have those soils and sites responsive, then we see basically a response to sulfur application. So to finish a little bit, I mean, we are trying to connect all this data with the contribution of fixation. So we have data on nitrogen fixation at the point of maximum fixation. If you remember from the beginning of the presentation, that was around pod formation. So if you look at this map, this is one of our first map available in terms of a star disclosing how much contribution of fixation is going to the plant. We have some places where the amount of nitrogen fixation is low, 34, 42%. We have in some locations in northern part of, I mean, we have close to 50, same is in some locations in Indiana and Ohio. So clearly we have a lot of variability. And this is important to think about understanding how this variability is connected in terms of nitrogen fixation. So in many situations, unfortunately for many, high yields are not really associated to more fixation, okay? There are in some situations that we are getting to very high yields, and it's also because we are using a lot of nitrogen coming from the soil, okay? So it's something that we really need to study. Fixation is becoming important, but also in many situations in high yield, the contribution of nitrogen from soil is very relevant. In overall, if you think about a magical number, as we usually want to get in many situations, we can say that the average fixation for across many of these studies was around 40%, which 40% of that total demand for soybean came from fixation. And lastly, to finish, which I think that is just thinking about more the future, when you're looking at that sulfur response and sulfur response connected mainly to the responsive yield sites, in a previous study, we also look very closer to the quality. We also have data on quality for this study, so that we will be exploring more in the next steps. And these sulfur effects are also connected in some situations when we see yield response to sulfur in that study. We also see a yield response to sulfur amino acids. So if you think about it, or at least if we start talking about quality, I think that it's important to recognize that this impact on sulfur might not only connect to yield, but also in this case, connected to very important sulfur amino acids, I mean, in terms of basically response to, I mean, increasing the concentration on quality. So summary, I mean, take home from this presentation is we still need to focus more on what are the best management, and the nitrogen seems to be very unlikely. If you think about terms of response to fertilization, we're trying to emphasize this point because there is always a question coming about, should I fertilize soybeans? And then if you look at all the data that we have collected, I mean, the last several years, it shows that it's very unlikely and even uneconomically. We also start quantifying these uncertainties. I mean, and uncertainties usually are large. So we really need to understand better what are the best diagnostic tools. And in going that direction, these changes on quantification of these nitrogen changes during the season really help us to understand a little bit more when are the situations where we can see response to sulfur. And mainly that yield response, as I mentioned before, was connected to sulfur application. And again, as I mentioned in the past, I mean, and also we were able to look at this study, when we were applying very small amounts of nitrogen early season, we haven't seen much of an effect on the fixation, which is important just to understand that very small amounts, they don't really do much in terms of changing that contribution from fixation. And then just to leave it as thinking for the future, that application of sulfur, and in many cases, I mean, in situations where we might not see a potential impact on yield, we might need to start thinking about the quality and then potential effects on that sulfur amino acids. So I think that I will stop here for this first part of the presentation. I know that we have Sean Castile from Purdue University, and then Eros Francisco from Auburn University. I mean, just to go through and then have a chance to do some Q&A session.
Fantastic presentation, Ignacio. Thank you so much. And we already have some questions coming into the chat for our panelists. So we'll go ahead and start with the first question. Can you comment on under what situations one should consider treating seed with inoculant in fields that have had a long history of soybeans? And I think our extension recommendations for this vary across the United States. So let's go ahead and start with Ignacio, and then we'll go to Sean and Eros on this one.
Yeah, that's an excellent question. I always say if you are feeling comfortable as a crop insurance option, just kind of do it. But we are seeing that in many of our soils where we have been having soybean in the past years, I mean, we don't get to see much response to inoculation. So if you really want to use an inoculant just to feel safe, I mean, use the cheapest one.
Yeah, in terms of Indiana Midwest, usually if we've been out of soybeans 3 to 4 years is when we go to inoculant as a need. In a lot of cases, if it's been longer than that, look at even a double rate or 2 modes of action in terms of seed treatment or in furrow are the options. If it's a typical corn soybean rotation on the soybean front, really we don't see any benefit from it. And as Ignacio said, if you want to do it as a $4 insurance essentially for what some of these inoculants cost, you can, but as far as the need, not unless it's been out for 3 or 4 years at least.
Well, here down in the south, there's no recommendation for like 2 years or 3 years or 4 years or every year. If you're growing soybeans for a long time, there is the feeling that you don't need to redo the inoculation. But I agree with the other panelists and with Ignacio. We really need to advocate for more inoculation because it's a cheap insurance for the crop. And well, we may discuss this in more depth in the other questions. There are many situations that we can kill the bacteria in different practices. So we got to pay attention to that.
Fantastic. So we'll move into the next question, which is what and when is the best application to increase protein with sulfur? And Sean Castile's done a lot of work with sulfur. So let's start with Sean and then we'll move to the other panelists.
Sure. Yeah. And in terms of the soybean response to sulfur, so I want to kind of piggyback a few things that Ignacio talked about. And we certainly do have locations that respond year in and year out to sulfur. Those are going to be the more classic coarse texture, but still somewhat low organic matter. But we can even see it on high organic matter, 4% type soils. If they're planted earlier, that tends to be the kind of that combination is that we have cool soils, limited mineralization. So you have limited sulfur, limited nitrogen supply. So that sulfur benefit that Ignacio talked about is affecting nodulation, and it's therefore is affecting nitrogen. It's affecting nitrogen supply through fixation. So just to make that clear, the sulfur response is an effect on fixation and ultimately nitrogen. So in terms of the protein, what's interesting on our work, on our very responsive sites, we'll see an absolute increase in protein on upwards of 2 to 4% protein. So if we go from a 36% dry matter, we'll go up to 30 dry basis, we'll go to 39, 40% protein. And then in some of the other scenarios, we'll see about a 1% increase, even if the yield increase is not as good. So we see that the when and the where aspect of that Most of all my recommendations is close to as planting as possible for that sulfur to affect nodulation and fixation, and then to cascade through on the seed remobilization and protein build.
Yeah. Yeah, if I need to add, exactly the same. I mean, Sean explained it very well. So that sulfur effect is on the fixation. So if you think about when to add it, as early as possible, because we need to start that fixation and make sure that that fixation is boosting that, supply of nitrogen to the plant. One thing that Sean also mentioned is situations where we haven't seen response in yield, but we tend to see sometimes even response in protein. And we tend to see increase in protein. And then one of the data that we are analyzing, we were analyzing a couple of studies, and we have available for this study too, is that it's interesting to start looking at the amino acid content. And sulfur amino acids, is something very relevant for pork industry and from other industries on animal side. So thinking about potential for increasing methionine or cysteine amino acid, is something for the future, looking at quality as also an alternative view of what is the overall impact on sulfur on soybeans.
The second part of that question we didn't answer was the what. In terms of looking at a sulfur source, look at soluble sources close to planting. So granular AMS, pelletized gypsum, those are great sources. You look at Mes10, that's half elemental sulfur, half ammonium sulfate. So you'll get half of that benefit. Elemental sulfur, it takes 300-400 days to fully oxidize. And so we're just getting into that space of, okay, do you have some residual effect? But those soluble sources are going to be the ones to go for.
Great. So we'll move to our next question. I'll throw this one to you first, Eros. What is the effect of late season nitrogen application on yield?
Well, we need to remember, and Dr. Ciampiti showed that the peak of nitrogen uptake occurs around or 3 or 4, depending on the variety or the cycle. And nitrogen is a macronutrient, so the plant demands a lot of nitrogen. So when we talk or when we mention about applying some nitrogen to a soybean crop, we're not talking about a huge amount of nitrogen, because it gotta be the soil, the main source of nitrogen for the soybean crop. So what we see is some folks are trying to apply, I don't know, 5 pounds here, 3 pounds there, just to give a boost in the crop, or perhaps a shine in the leaves. But that amount is not going to create yield per se. That amount is just momentarily helping the plant, but is not increasing the yield per se. So I never advocate for applying nitrogen, a big amount of nitrogen, for a soybean crop. And Ciampiti showed that, and I have a lot of experience back in Brazil when I was there. We had 37 field trials across the entire country, and only 5 sites showed response to nitrogen application. And those 5 sites were demanding nitrogen for any crop, not just for soybean. So we got to be careful when we see this type of literature. So what are the conditions that are favoring a nitrogen response for soybeans? It might be the same conditions for corn, for cotton, for peanuts. So not only for soybeans.
And maybe to add to that, we have done studies in the past looking at even historical varieties, and we did inoculated versus, we were applying 30 pounds at R3, and we haven't seen much effect. And then my frustration, when I discuss with farmers, is that if I cannot give you a certainty, a more certainty in the responses, I will just not recommend it. If I see one response in 100 locations, my recommendation is not do it. And I think that I agree with Eros in many of the concepts on look at the amount of nitrogen that the plant needs, and look at that demand over time, and how big is that demand as you are creating more bushels. That 30 pound is only helping alleviate the plant just to change the status. But if we have a problem of fixation, and as we were mentioning, that fixation seems to be the main source at the end of the season, because we deplete all the soil nitrogen. Most of the nitrates are depleted around mid-August. So unless the plant is under really good conditions, we really need to focus more about canopy health and making sure the plant is under well-balanced nutrition, to make sure that fixation is kicking and working as the best efficiency possible.
And if I may add another comment before Sean, last year, I forgot to mention this. Last year, I tested 100 pounds of urea. So we are talking about 45 pounds of nitrogen per acre, and I didn't see any response. So in a very sandy soil, in a coastal plain type of soil. So.
Great. We've got a lot of questions, which is great. So I'm going to throw the next one to Sean. So using sulfate as a sulfur source, which we did in some of our collaborative work, do you think there was any response to calcium?
So they're referring to calcium sulfate in that situation. So with that, I don't think calcium's the driver in this. I think when you do see the responses, it is sulfur based, so sulfate. So as I put it, sulfate is the mobile form, is the plant available form. And so that is what it's responding to now, whether that's getting it through calcium sulfate as pelletized gypsum, or if they're getting it through ammonium sulfate. Any form to get that. Even another one that wasn't mentioned today, ammonium thiosulfate. That has to get oxidized even temporarily, the 2-3 week time period, but it eventually gets to the sulfate form, which is going to be taken up. The calcium aspect of this, I don't think that's the play in terms of these yield responses or protein changes that we've been describing.
Can I add something to that, Rachel? I have done this, and this is crazy. Just for research, we had applied more than 10 tons of gypsum per hectare, and we check it against single superphosphate, which is a phosphate with calcium, and we didn't see any difference between those. So if calcium was the main driver for boosting soybean yields, that would be seen in the test, and we didn't see this.
Great. I'm going to combine the next two questions, and they're in regards to new strains of inoculant that are available on the market. So have you tested these different strains, and how does that impact your recommendations?
I can start if you want with, I showed that combination of rhizobium with azospirillum. Most of the idea why we wanted to test that is because we have seen many researchers from Brazil, and Eros is probably familiar with some of this research, that they are seeing responses. When they combine different microorganisms. Our data shows that in many situations we are not seeing any effect. When we're combining rhizobium and azospirillum, when we are looking at other inoculants with other strains, we also did some testing. The challenge is if the plant is already colonized by a specific strong type of microorganism, even if you introduce a different one, it was not going to change much on the equation. So if you really are focusing on thinking about how inoculants can help soybeans, we need to really kind of think about how we reformulate the inoculation, and maybe thinking about if we can develop more kind of better inoculants, or strong microorganisms so they can help to boost that fixation. But overall, there is a cost on the fixation, and then those bacteria, they usually also lose nitrogen. There is a part that they basically denitrify, they lose nitrogen to the atmosphere. So there is an efficiency that we might probably be able to help and increase, but I haven't seen much, even when looking at different inoculants on the market, I haven't seen much difference, between yield productivity so far.
Right, and I'll echo in terms, I think the question was relating to the two different Brady rhizobium in that, and I don't think we've been seeing anything. South America might catch a few and a little bit on the second strain, but I have not seen anything with the products we've got available here.
Okay, I'm going to move into the next question. We've got a few more to move through, so I'm going to have to skip through a couple, but the next one, in the Dakotas and Minnesota, excessive carryover nitrogen increases IDC significantly, iron chlorosis. How about your areas?
Well, it's a quick answer for Purdue or in Indiana and Illinois. We really don't have to deal a lot with IDC, so it's not an issue for us. But in terms of what you see, yeah, I mean, that's pretty part and parcel from what I hear from colleagues up north.
Yeah, same on our side. I mean, if you want to jump quickly with Sean, I mean, we don't see so much problems on IDC, and also what I will say is based on the research that we're conducting, we're not seeing that much of a carryover.
Right.
Okay.
We don't have much IDC in the south, but the black belt region only.
All right, so the next question is, a lot of non-AMS water conditioners get used in our area when spraying pesticides. Would switching back to AMS as a water conditioner make any real difference in terms of foliar sulfur provided to the plant?
No, I'll go ahead and jump in on this one right away. I've done foliar S work, and to get to the rates that are going to see anything on fields that are responsive, we need to be close to 3 pounds of sulfur per acre. And the rates that they're talking about back and mixing with herbicides, they're nowhere near that. And they're just going to be a blink in the wind kind of thing. There's hardly going to be enough there to reach any of those kind of needs.
I would say it's the same concept for all macros. We cannot apply too much macronutrients via foliar. So,
yeah, we always need to remind to everyone that the roots are basically the plant fractions that they are supposed to absorb nutrients. So the absorption coming from foliar is very inefficient.
Well, yeah, I'll finish my statement on foliar. So we did foliar versus granular application, and we did get the response on responsive sites, but it still was not to the same level of what our granular application was at planting. Again, these are kind of rescue foliar treatments of V4. So you're a month and a half after you've already had granular AMS out there in this example, and it's just not going to yield the same in that comparison.
I just want to add another comment. And we have a minimum or a critical level for sulfur, right? That we tend to see result or responses if we are under this critical level for sulfur. But sometimes what I see is we have sulfur in the soil, but we need to understand that sulfur is a little bit leachable in the profile. So we may find sulfur out of the root zone in the soil. But in some cases, the root is not growing enough to reach where sulfur is. And that might be because of soil compaction, because of acidity, or any other problem that is not allowing the root to grow enough to pick up those the sulfur that is in the profile, but not in the root zone.
Great, and I think that kind of answers the next question, Eros, about critical soil test levels for sulfur, and any comments in regards to specifically low organic matter or coastal plain soils. Does anybody want to add anything to that?
I did spend 8 years in North Carolina, so we would see some sulfur deficiencies, but then you'd hit like a BT horizon, 16, 18 inches, coastal plains there, and then they'd be fine. So again, it's about some of the soils itself. And if it had subsoils that are more acidic, the sulfate would be more prone to stay there than leach through when you have those kind of subsoils. So that's kind of that part and parcel of situation where you can see some transient sulfur deficiency and then go away whenever it reached a zone where it was accumulating.
If you are in a zone where you don't have that BT layer, but you have all sandy soil through the profile, you may be losing sulfur. So the advice would be put sulfur whenever you have opportunity for every crop, not just soybeans. Try to keep organic matter in high via cover crops or any type of manure that is available in the region. That would be my advice.
And I think that this attendee was also trying to interconnect this question back to decision-making about inoculation in relationship to a certain sulfur test level. So any comments on that?
If you have low organic matter soil, so again, low is relative, right? I grew up in flat black Illinois soil, and low is 3.5%, but that's not low. right? Less than 2%, less than 1% organic matter, coarse texture soils, CECs that are going to be 3 and 4. Those are going to be fields that are going to be primed for, okay, that sulfur application to help with nodulation. They're going to probably be primed for more than inoculant situation. Yep. So then to make sure that you do have a good nitrogen supply. If you're sold short on nitrogen via fixation or organic matter, you're sold short on yield potential. So I would definitely think that those situations would be more prone to say, yes, let's go inoculate. Yes, let's go and look at the sulfur as a potential responsive site.
Yeah.
Great. I'm going to move into the next question. So for a number of years, there was a perception for each bushel of soybeans harvested, your land was left with 1 pound of usable nitrogen. Lately, I've seen many articles where you cannot depend on any nitrogen left from the previous soybean crop. And our Science for Success group has actually been talking about this and trying to do a project nationally to update our recommendations. But what information is accurate? Does anybody want to make any comments about this?
Well, I can jump on maybe one of the slides that I use. I mean, if you think about what is the overall need of nitrogen from soybeans, and if you look at the overall nitrogen budget, no, I mean, we need to probably, there is a perception that we call the end credit, and then we need to be careful about how we use the term end credit, because the credit means, end credit doesn't mean that you are gaining nitrogen. It's just that the nitrogen is more available for the following crop. And it's just a connection with the carbon nitrogen residue from the soybeans, that nitrogen will become more available for the following crop in the rotation. But that doesn't necessarily mean that we are adding or leaving nitrogen in our systems. There are situations, and we have numbers now, for example, that every time that we have fixation levels below 30%, or even in situations that we have some of our levels here that we quantify that they are below 50 or 60%, we know for sure that we are using more nitrogen coming from soil than the one that is coming from fixation. And in most of those situations that you are using more nitrogen coming from soil, it's clear that that rule of leaving behind nitrogen doesn't apply. And so far we haven't got, or at least we have still probably not enough information or sufficient information just to quantify and to put a number to say how much nitrogen is coming from roots, what is the overall contribution. But we know that in many situations from the fixation side, it might be maybe 20, 30 pounds extra that can help to become that probably balance more close to zero. But that misconception of the credit is not really that we're leaving nitrogen behind. It's mainly that nitrogen that is available present on soybean residue is becoming basically more rapidly available for the following crop in the rotation.
Usually that's used in line when I think of a corn soybean rotation. How much more nitrogen do I need for the corn crop?
Exactly.
Continuous corn situation, you're 30 to 40 units more because you flat don't have it, versus corn soy, their rotation, the nitrogen recommendation is going to be less. So we've often said it as a credit, but really what it is if you got corn after corn, high carbon content, not much nitrogen being supplied, you have to apply more. So that number is going to look almost identical to most extension fact sheets, 30-ish nitrogen pounds, right? But it's not a credit, it's just you need more in a high carbon environment.
Yeah, and if we might need to change that to call it more a cropping system effect.
Yeah.
Great. I'm going to move into the final question for the panel. It says, Ignacio, you mentioned that M fixation can be reduced in high organic matter soils and high clay soils. I understand the end release from organic matter, but not the clay content. Can you explain?
Yeah, clay usually, if you think about, I mean, when you have a high clay content, it can act as a protection to the organic matter. So we have many pockets in the soil where high clay content that organic matter is more protected, which means that, again, nitrogen is coming from organic matter. But when we have organic matter that is a little bit more protected, that nitrogen is the release of nitrogen from organic matter can be quite different. And what we are trying to understand with some of the information that we are presenting today is how that mineralization works. Because at the end of the day, if we really want to understand the fixation, it's clear that we cannot understand fixation without understanding the mineralization of nitrogen in the soils. And in those cases where we have high clay, we tend to have high organic matter and it's more protected. And that or nitrogen usually comes later in the season when the plant is fixing. And then usually there is that kind of a competition between that nitrogen that is available present in the soil with the amount of nitrogen that the plant is receiving via fixation. So in many situations, as we show in one of the graphs, when we have a peak of mineralization that it comes late in the season, what finish doing to the plant is finishing impacting the overall fixation potential. And that's one of the things that we need to start at least to investigate a little bit more to see what are the things that we can do in terms of management. One potential management is to make sure that we are planting early in many of those studies because we don't want to have that peak of mineralization coming that late, more when it's connected to the high fixation demand. So that's the relationship between the high clay soils. I mean, they're basically having more organic matter and it's a little more protected, and then that nitrogen is being released late in the season and it's impacting more that potential for fixation.
Fantastic. Well, our time for this webinar is coming to an end. We'd like to thank our panelists, Dr. Eros Francisco, Dr. Ignacio Campiati, and Dr. Sean Castile, and all the attendees that have plugged into this webinar series from all across the United States. We've collected the data. We have a strong presence from the Midwest, the Mid-South, the Southeast, and even Canada attending these webinars, which is really exciting. You will be getting a survey for those of you who have attended these webinars, as well as a link to the webinar recording and the Science for Success developed resources that have been shared in these webinars. So please fill out the survey and provide us some feedback on topics you'd like to see covered in future Science for Success webinars. And we hope everybody has a great weekend.